That can be bacterial.
Yeah, it's burned and then something else comes in.
That's a problem.
This fall, NMSU students studying plant disease diagnostics gained real-world experience by offering their services to the public. Dr. Soum Sanogo's class held a plant diagnostic clinic at the Las Cruces Convention Center. Everyone from weekend gardeners to professional growers brought samples for students to examine and received advice on how to treat various plant disorders.
Well, in the classroom, we do a lot of different examples, and we're studying the abiotic and biotic stresses that, when applied to a plant, create a disorder. So what this does, it provides us an actual hands-on experience to see just a random crop or plant, and then to verify and go line by line. Well, is it abiotic? Is it biotic? Is it a discoloration? It actually allows us to use what we've been learning. So it's not so much a test as sort of a bigger lab, it's like an application.
What we try to do is to teach the student not what to think, but to teach them how to think critically so that they can approach a problem and go about gathering all the information so that they can solve that problem.
Students learn how to diagnose plant disorders by using the W3H2 approach. The 3 W's stand for what, where, and when, while the 2 H's stand for how much and how.
Well, when they bring us a sample, we're told to ask 5 or 6 questions, and it's like, when did it happen? Where did it happen? What made it happen? How long has it been happening? And how bad is it? And we look at their symptoms, and then we decide whether it's environmental, from the sun, water, nutrients, or if it's from a pathogen or a bacteria or an insect, and the different symptoms. And then we ask them, how's it been watered? How's it been treated? Full sunlight, what are the environmental conditions that it's in? And then from there, we kind of go through a list of things and cross them off as we get the answers, and then give them a diagnosis or some way of correcting the problem.
In using the concept of W3H2, the diagnosis progress is separated into what Sanogo calls 3 orbits. The first orbit is to categorize the disorder. The second orbit is to determine the causal agent.
So in this case, we cover pretty much all possible causes of disorders. They can be the so-called biotic, meaning they are caused by living organisms, or they can be abiotic, meaning they are caused by non-living.
The third orbit is to determine which approach needs to be taken.
And in here, it helps to recognize what I call micro and macro approach. So this teaches the student to take all the information of a category, causal agent, and putting them together and looking at them at different scale depending on the scenario.
After making an initial diagnosis, students will examine samples of the diseased plants in the lab.
Like in this case, we're looking at the leaf spot, which in this context are caused by bacteria. So the student will ask to make cuts out of his symptomatic leaf, mount them in the microscope, and observe for the oozing of a bacteria. And another sample here, which is a pod rot in peanut, the student will mount small pieces from the shell or the seed, and again proceed to do microscopic observation and see if they're able to see any organism at this point.
Students perform microscopic observations on the samples. However, whatever they see may not always be the causal agent, so the sample is prepared for isolation. Then they apply 4 criteria known as Koch's postulates.
And Koch's postulate really is a set of rules that allow you to indicate or to confirm whether an organism that you have isolated from, say, this peanut pod is in fact causing the disease that you have observed.
In the past, work outside the lab and classroom has included a trip to a farm field. This is the first time Dr. Sanogo's class has organized a plant diagnostic clinic designed to help multiple growers.
It gives the students an excellent opportunity to come out here and go through this diagnostic process and try to figure out what's going on with these different landscape and ornamental plants, and sort of put this knowledge that Soum has given them over the course of this semester all together and figure out something from start to finish here, and then provide these folks that brought in the plant samples back with a report so they know what was going on.
Because we do know we have a lot of people that are growing plants here, maybe for the first time in the Southwest. And it's a lot more challenging than just putting a plant out there and watering it, and hoping to reap the benefits. So you run into a lot of problems with insects, disease, soil problems that you're not aware of.
It's very helpful to the students to come out and do this, but also you're getting knowledge from the students, but like I said, you're getting knowledge from people that are top of their fields from the university that may know more than like your local nursery grower may know.
Students will send their final diagnostic reports as soon as lab work concludes. The event has proven to be mutually beneficial for both students and the community. For plant disease diagnosis, please visit your local county extension office and watch for more clinics like this one to be held in the future.